Welcome back with the continuation of our main topic, lipids. Here, in this video, we are going to talk about phospholipids and sphingolipids. Previously, we have discussed lipids and its two main types that were named as hydrolyzable and non-hydrolyzable. Now, let's begin with our main topic of the day. Here we are going to discuss fats, phospholipids, sphingolipids. Let's start it with fats. Fats, or triacylglycerol, are esters of glycerol with 3 fatty acids. Within the cell, they mainly occur as fat droplets. In the blood, they are transported in the hydrophobic interior of lipoproteins. When a single fatty acid is esterified with glycerol, the product is referred to as a monoacylglycerol. Formally, esterification with additional fatty acids leads to diacylglycerol and ultimately to triacylglycerol, the actual fat. Interesting point to bring into your attention is that triacylglycerol was formerly termed as triglyceride, which is now commonly known as triacylglycerol, so don't confuse it. Triacylglycerol is the correct chemical name, whereas triglycerides is the common name for the substance. As triacylglycerols are uncharged, they are also referred to as neutral fats. The carbon atoms of glycerol are not usually equivalent in fats. They are distinguished by their SN number, where SN stands for stereospecific numbering. The 3 acyl residues of a fat molecule may differ in terms of their chain length and the number of double bonds they contain. This results in a large number of possible combinations of individual fat molecules. When extracted from biological materials, fats always represent mixtures of very similar compounds, which differ in their fatty acid residues. Now let's revise a bit of our previous knowledge and understand the concept of chiral compound. Chirality is an important geometric property relating to a molecule's symmetry. Chirality means asymmetric in such a way that the structure and its mirror image are not superimposable. Chiral molecules are always asymmetric at one or more centers. You can see here shows the simplest example. Right and left hands are non-superimposable mirror images. It represents chiral compounds, whereas achiral compounds are the superimposable mirror images. You can see the example of bottle here, which can easily be imposed on one another. A chiral center can arise at the middle carbon atom of a triacylglycerol if the 2 external fatty acids are different. The monoacylglycerols and diacylglycerols shown here are also chiral compounds. Now let's discuss phospholipids. Phospholipids are important components of cell membranes and lipoproteins. They are amphipathic compounds, which means they have an affinity for both aqueous and non-aqueous environments. The hydrophobic part of the molecule associates with hydrophobic lipid molecules, while the hydrophilic part of the molecule associates with water. In this way, phospholipids are compounds that form bridges between water and lipids. The parent molecule of the phospholipid family is phosphatidic acid. It consists of a glycerol backbone to which are esterified 2 fatty acyl molecules and phosphoric acid. The latter produces a phosphate, which is free to react with the hydroxyl groups of serine, ethanol amine, or choline to form phosphatidylserine, phosphatidylethanolamine, or phosphatidylcholine, respectively. Well, phosphatidylcholine, or lecithin, is the most abundant phospholipid in membranes. This is commonly known as lecithin and is frequently used in food as an emulsifying agent, whereby it causes lipids to associate with water molecules. Now let's discuss RDS. Respiratory distress syndrome, or RDS, is a common problem in premature infants. The immature lung fails to produce dipametoyl lecithin, which is a surfactant. RDS occurs when the alveoli collapse inwards after expiration and adhere under the prevailing surface tension. This is atelectasis. Atelectasis is a complete or partial collapse of the entire lung or lobe of the lung. It occurs when the tiny air sacs, alveoli, within the lung become deflated or possibly filled with alveolar fluid. The function of dipametoyl lecithin is to reduce the surface tension and permit expansion of the alveoli on inflation. Assessment of the maturity of fetal lung function can be made by measuring the ratio of lecithin to sphingomyelin in amniotic fluid. Next, we have sphingolipids, which are found in large quantities in the membranes of nerve cells, in the brain and in neural tissues. Have a slightly different structure from the other membrane lipids discussed so far. It is a complex lipid that contains sphingosine rather than glycerol. It consists of sphingosine to which fatty acid, phosphate group, and choline are attached. You can see this here. Sphingosine is an 18-carbon amino alcohol with an unsaturated hydrocarbon chain. It is the backbone of sphingolipid. When sphingosine forms an amide bond to a fatty acid, the compound is called ceramide. This is the precursor of the sphingolipids. Sphingomyelin, the most important sphingolipid, it has an additional phosphate residue with a choline group attached to it on the sphingosine, in addition to the fatty acid. Sphingolipidoses are a class of lipid storage disorders or degenerative storage disorders caused by deficiency of an enzyme that is required for the catabolism of lipids. Gaucher's disease is both a sphingolipidosis and a lysosomal storage disorder due to a specific deficiency in an acidic glucose cerebrosidase that results in accumulation of glucosylceramide, primarily within the lysosome. Gaucher's disease, the most prevalent lysosomal storage disease, is an autosomal recessive disorder caused by lysosomal deficiency of beta-glucocerebrosidase, or GBA. This results in excessive accumulation of glucocerebroside in the brain, liver, bone marrow, and spleen. Type 1 Gaucher's disease, or non-neuronopathic form, can be treated by enzyme replacement therapy with recombinant beta-glucocerebrosidase. In the future, Gaucher's disease is a potential candidate for gene therapy by inserting the GBA gene into hematopoietic stem cells. Lastly, let's get an overview of glycolipids. Glycolipids are present in all tissues on the outer surface of the plasma membrane. They consist of sphingosine, a fatty acid, and an oligosaccharide residue, which can sometimes be quite large. The phosphate residue, typical of phospholipids, is absent. Galactosylceramide and glucosylceramide, which are also known as cerebroside, are simple representatives of this group. Cerebrosides in which the sugar is esterified with sulfuric acid are known as sulfatides. Gangliosides are the most complex glycolipids. They constitute a large family of membrane lipids, with receptor functions that are as yet largely unknown. A characteristic component of many gangliosides is N-acetylneuramic acid. That's all of this topic. For more knowledge stuff videos, keep watching sqadia.com.